Hi, welcome to AP Biology. I'm Ms. Kaufman, and this is the first of a series of videos that's going to help guide you through the content of AP Biology. I hope that when I ask you questions in these videos that you take the time to actually try to answer them, because the more that you put into this, the more the brain work that you do, the more you'll get out of it. So let's go for it. This is Unit 1, Chemistry of Life. Let's start our review of chemistry by looking at an atom. So what is an atom, and what are the three subatomic particles in an atom? An atom, by definition, is the smallest unit of a chemical element that retains the properties of that element. It contains three subatomic particles: protons, neutrons, and electrons. Now we can represent an atom visually, like we see in this picture of an oxygen atom on this slide. A visual representation is an example of a model. A model is just a way of representing a concept or a physical object in a way that simplifies it. And that's one of the great things about a model, is that it makes something that's complex simpler. But it's also one of the dangers of a model, in that every time you simplify something, you lose some of the truth of it. So whenever you look at a model, whether it's a picture or a 3D model or a mathematical model, I want you to think about what's wrong with the model and also what's helpful about the model. So in this case, what's wrong about this model of an atom and what's useful about it? And also, how else could we model an atom? So one of the things that I think this model does well at is showing us those three subatomic particles. So we can see that the protons and neutrons are found in the nucleus, and the electrons are outside of the nucleus. Something that it doesn't do very well at is showing us scale. In reality, electrons are much, much smaller than protons and neutrons, and they're actually much farther away from the nucleus, and they're not in fixed positions. They're whirring around all over the place. So those are some of the things that are good and bad about this particular model. You might have found it even more. Other ways of modeling an atom, this is kind of infinite as well, but you could model it sort of in a 3D model. You could have a computerized model of it. You could have a mathematical model. These are all different representations of an atom. All right, so here's a mini periodic table. I want you to be familiar with some of the elements that are most common to living organisms. And so those include hydrogen, carbon, nitrogen, oxygen, sodium, magnesium, phosphorus, sulfur, and chlorine. So if you see those symbols, like Mg, for example, capital M, lowercase g, I need you to know that that's magnesium. You don't need to memorize the atomic mass of these, but I do want you to be familiar with those symbols. I also want you to understand why the periodic table is organized the way it is. So let's take a moment and just let you remember, how is the periodic table organized? Why is it set up this way? Well, one of the ways that the periodic table is organized is based on the number of protons. So the atomic number for each of these elements is corresponding to the number of protons. So for example, hydrogen has 1 proton, helium has 2, lithium has 3, and so the elements get larger as you move to the right and then as you step down in the rows. This is also related to some other properties of the elements. As they gain protons, if they are neutral, then they also are gaining electrons. So carbon, for example, has 6 protons by definition. If it has 6 protons, it's carbon. If it's carbon, it has 6 protons. But if it's a neutral atom of carbon, that must mean it also has 6 electrons, because neutral means it has no net charge. So the number of protons must be equal to the number of electrons. So a neutral atom of carbon has 6 protons, 6 electrons. As you move to the right, you will then have more protons, and also in neutral atoms, more electrons. This relates to how full the valence shell of the electron valence shell is. So for example, in that first row, the first shell can hold 2 electrons. And so when you get to helium, that shell is full. No more electrons can fit in that area. Then when we step down a row, there's room for 8 electrons. And so the farther we go to the right, the more of those 8 slots essentially are filled up. So neon, all of those slots are filled up, and it's a stable atom. Whereas fluorine, F, is just one away from having its valence shell full, and so it really would like to have one more valence electron. And I'll make a note here that I often talk about, I sort of personify atoms and all sorts of molecules and things. It's not really correct, but it absolutely helps me sort of understand what's going on. But it is something to be aware of, that these atoms don't actually have feelings. They don't actually sit up at night and dream of electrons, right? But we can talk about this to sort of help understand the trends. All right, so one trend that's really important is electronegativity. So electronegativity measures an atom's tendency to attract electrons. Electronegativity trends relate to valence electrons, just like I was saying, and the electronegativity differences relate to bonding. So right now, take a moment and make some observations based on this slide. What do you remember from previous exposure to chemistry, or what's new here? Make some observations based on this diagram, or these diagrams. All right, there is so much to look at here, but one thing you might notice on the bottom right is a periodic table where the colors correspond to electronegativity values. And you notice in the very right column, those are the noble gases, and so those have their valence shell completely full, so they have no electron. They have no desire for additional electrons. Whereas the darker the red the color, the higher the electronegativity, the more they want to attract electrons, or the more those atoms attract electrons. If you have one atom that has a really high electronegativity, paired up with an atom that has a very low electronegativity, that's going to be a very big electronegativity difference. And you can see that modeled in the ionic bonding, where there's electrons completely transferred. So in this case, the little blue atom pulled so hard that the string broke and it got all of those electrons. So those two atoms are not shown sharing those electrons. There is a complete transfer. So this is ionic because these are now ions. These are now charged particles. The blue atom here that got extra electrons is now extra negative. So that's a negative ion. And the purple one that lost its electrons is now less negative, or it's a positive ion. Above that, you can see a polar covalent bond being modeled. In this case, there's a difference in electronegativity, but it's not different enough for the electrons to be completely transferred. So they're still technically sharing those electrons. It's a covalent bond, but the sharing is not equal. So that blue atom is pulling stronger, which means it has, the electrons are closer to it. So it has a partial negative charge, but not a full transfer. So it's not an ion, it just has a partial negative charge. You can also see this modeled on the left side of this slide with those sort of blue diagrams. So a covalent bond formed between identical atoms, the electrons are going to be shared perfectly. So for example, if you have two atoms of oxygen, even though both of those oxygen atoms have relatively high electronegativities, if it's an oxygen versus an oxygen, they're going to share those equally. A polar covalent bond is modeled in the middle of those blue diagrams, showing that there is a partial charge. So this is a covalent bond. They're sharing the electrons, but there's a partial charge. And then an ionic bond, there's no electron sharing because there's a complete transfer. All right, let's dive into the three most important atoms in biology. And I would claim that these are the three most important atoms in biology because they appear in almost every single molecule in our bodies. So we need to know a bit about these atoms. All right, so first of all, hydrogen. Hydrogen typically forms one bond. Then we have oxygen that typically forms two bonds, and carbon that typically forms four bonds. Let's take a moment and see if you can figure out why that might be. Why does hydrogen form one? Why does oxygen form two? Why does carbon form four? So this has to do with how many electrons or how many extra spots in the valence shell there are available for additional electrons. So hydrogen has one extra spot in its shell. Oxygen has two extra spots in its shell. Carbon has four extra spots in its shell. So oxygen, for example, has 6 valence electrons, which means it has room for 2 more. That means it can form 2 bonds because those are areas where electrons can essentially kind of fit in. Carbon has 4 valence electrons, and so it has room for 4 more to create a full outer shell of 8. Carbon can form those 4 bonds in all sorts of different ways. So it can form 4 single bonds where it's bonding to 4 different atoms, or it can form double bonds where it's 2 of those connections are with the same atom. It can even form a triple bond where it has 3 bonds, so 3 pairs of electrons are being shared with a single other atom. In the oxygen, you notice that it says 2-. So oxygen, if it forms an ion, that would mean that it gained 2 additional electrons to fit into its shell. And so then that would be in ionic form, right? So that's showing as an oxide ion is what you have there. It's oxygen with 2 additional electrons. If we move on to hydrogen, so for whatever reason, I'm moving right to left. Hydrogen, you can see it has 1 proton and 1 electron in its neutral state. That hydrogen will often lose that single electron. If it does, it becomes positively charged because it has less negativity, right? And so if it has less electrons, it only had 1 to begin with. So if it loses that 1 electron, has no electrons left, which means it's just a proton. So hydrogen ion, an H+ ion, is also referred to as a proton, and that's why. All right, we can represent chemical structures in a whole lot of different ways. Take a look at these 4 different methods of representing the very same molecule and see what are some advantages and disadvantages of each method. So as we move from left to right, we sort of have more simplistic, which means we're going to lose complexity. So an advantage of the one on the left is that it has all sorts of information. It's showing the bond length, it's showing the angle. Those funny symbols with like the dark triangle is and the slashed kind of lines are showing the 3D dimensions of those hydrogens poking into and out of the screen. So there's a whole lot of information here. As we move to the right, we have less and less information provided. An advantage of that is that then it's simpler. And simpler things are often easier to understand on sort of a big picture because then we aren't overwhelmed by additional information. So on the right, this is a skeletal structure diagram, and you are responsible for being able to represent molecules using a skeletal structure. So in this case, carbons are not drawn, but are at the unlabeled bends and ends. So there are going to be 3 hidden carbons here. 1 at each of those ends, and then 1 at the bend in that sort of intersection, right in the center of that drawn molecule that's double bonded to oxygen and single bonded to each of the other hydrogen, or each of the other carbons rather. Hydrogens are not drawn, but are attached everywhere needed to give carbon 4 attachments. So remember that carbon, because it has 4 valence electrons, has room for 4 additional bonds. And so every carbon is going to have 4 bonds. So you take a look at each carbon and you say, all right, how many bonds are used up by being bonded to other molecules that we know of, like oxygen or carbon? And then whatever's left is hydrogen. So if you compare the skeletal structure diagram to the one just to the left of it, it might help you understand where those hidden carbons are and where those hidden hydrogens are. All right, let's talk about one of the most important molecules of life. Now, this is not technically a biomolecule. It's not a carbon-based molecule, but it is incredibly important. So let's look at water. So what I want you to do is draw 2 molecules of water and label the intramolecular and intermolecular interactions. So just do this on a separate piece of paper, and then also label some key vocab of polar covalent bond, hydrogen bond, and polar molecule. All right, so here's one molecule of water. Let's just draw this for a moment, or look at this. So we have 1 molecule of oxygen and 2 molecules of hydrogen, which is why water is H2O. That subscript 2 on the H tells you that there are 2 hydrogen atoms in that molecule. So each of those hydrogen atoms are bonded to the oxygen. The bond between hydrogen and oxygen don't have the same electronegativity. Oxygen has a much higher electronegativity than hydrogen does, and that's important to know. So oxygen, even though this is a covalent bond, the oxygen and hydrogen are sharing electrons, they're not sharing them equally. So this is a polar covalent bond. That then tells you how two molecules of water are going to interact, because that partial positive on the hydrogen is going to interact with the partial negative of an oxygen on a different water molecule. So there is an attraction between the partial positive and the partial negative portions of these molecules. That attraction is called a hydrogen bond. So what we talked about earlier, the hydrogen to oxygen connection within the molecule, is polar covalent. The intermolecular bond, the bond between two molecules, is a hydrogen bond. And that's the attraction between the partial positive and partial negative portion of those molecules. Key root words here: intra means within, so intramolecular, within the molecule. Inter means between, so between two different molecules. So you can tell that there are two separate molecules here because there's no covalent connection between these two molecules. You need to have a covalent connection between molecules or you need to have a covalent connection between atoms for them to be part of the same molecule. So we have two molecules of water here. I should point out that the polar covalent bond is different than saying it's a polar molecule. So the polar covalent bond refers to just one oxygen to hydrogen connection within a molecule, and that's an uneven sharing of electrons. Saying it's a polar molecule means if we step back a little bit and we look at the overall structure of this molecule, there's a slightly negative side and a slightly positive side. So the molecule as a whole, because of those polar covalent bonds, is a polar molecule. All right, so now we have some more drawing. So I want you to draw out some properties of water. I want you to draw out cohesion, adhesion, surface tension, and then I also, on the next slide here, want you to draw what does it mean to be a solvent, what is specific heat, what is density, especially as it applies to water, and what is evaporative cooling. So I'm not going to go over all of these right now. Instead, we're going to, I want you to look at other videos made by other people on properties of water and use that to apply here. We're also going to do a properties of water lab where you will be more familiar with some of these. Additionally, you can find definitions for all of these terms on the Quizlet. And so I'll make sure that you have that link. But so all of the definitions can be found in your textbook, but also on Quizlet. All right, let's talk about how salt versus sugar behaves in water. So salt is an ionic molecule. So the connection between sodium and chlorine is just an attraction between a positive ion, which is the sodium, and the chlorine ion, which is the negative ion. So what happens here is this completely dissociates in water, and the polar water molecules surround those ions of sodium and chloride in a way that the partial negative portion of water, so the oxygen, surrounds or points towards the positively charged sodium ions, and the partial positive of the hydrogens in water surrounds the negative ions of chlorine. And you can see that in the diagram. Now, sugar is different. The intramolecular bonds in sugar are covalent bonds. So covalent bonds don't just fall apart in water like ionic bonds do. Okay? So covalent bonds stay together. So each individual sugar molecule is going to stay intact, and that's what's demonstrated with those little purple molecules there. The individual molecules stay together, but the molecules separate from one another. So if you have like a sugar cube and you put that in water, when it dissolves and you can no longer see it, it's not that any individual sugar molecule broke up, it's that all of those molecules moved apart from one another. Sugar and water are both polar, and so water is going to surround one molecule of sugar. All right, let's review molarity. So the first question I have for you as practice is to determine how much sucrose is needed to make 3 liters of a 0.2 molar solution. If you need a hint, you should use that molecular diagram in the upper right and also a periodic table. So the first step to do this problem is to determine sucrose's molecular mass. So to do that, we need to know sucrose's molecular formula. So the molecular formula is C12H22O11, and you should be able to get that from that diagram in the upper right, even though a lot of those carbons are hiding. So once we know the molecular formula, we can then figure out the molecular mass. There are 12 carbons, and carbon is 12 grams per mole. There are 22 hydrogens. Hydrogen is 1 gram per mole. There's 11 oxygens at 16 grams per mole. Put that all together and you get 342 grams per mole. Now that we have that information, we can determine the molarity. So we take 3 liters times 0.2 moles per liter, because that's what molarity is. Molarity is how many moles for every liter. And if we set it up in that way, then the liters will cancel. Then we multiply them by the molecular mass. Once again, that's going to allow the units to cancel. So we have moles on the top and moles on the bottom. Those will cancel out and leave us with grams. So 3 liters times 0.2 moles per liter times 342 grams per mole gives us approximately 205 grams. I'm not going to focus on significant figures right here. This is just approximately 205 grams. All right, what we have here is a picture of water. I want you to see if you can make any observations about this picture of water. If you look carefully, this is kind of like a Where's Waldo game, you'll notice that not all of the molecules pictured are water. So we have hiding in here a molecule of H3O plus and a molecule of OH minus. So the way we got H3O plus is if an H plus ion joined onto that water molecule. And the way we get OH minus is if an H plus ion left that water molecule. So water, pure water, has 10 to the negative 7 molar H3O plus ions, and it also has 10 to the negative 7 molar OH minus ions. That means in a liter of water, there's 10 to the negative 7 moles of H3O plus and 10 to the negative 7 moles of OH minus. OH minus is also called hydroxide. So what does this have to do with anything? Well, this relates to acids and bases and pH. So acidic solutions have a high concentration, and those brackets represent concentration. Acidic solutions have a high concentration of hydrogen ions. And neutral solutions, the concentration of H plus is equal to the concentration of hydroxide. Basic solutions have a low concentration of hydrogen ions. So why is that? Why is it that if there's a high concentration of hydrogen ions, that's a low pH number? And then the opposite. Why is it that basic solutions that have a low amount of hydrogen ions have a high pH number? So the reason that has to do with how pH is measured. pH is the log of the concentration of hydrogen ions. You don't actually have to make those calculations, but you do need to understand this general concept. So let's have an example. Let's actually start with our neutral solution in the middle, and we see that neutral solutions we know have 10 to the negative 7 molar H plus ions. So if we write that out, that's 0.00000001. Right? That is equivalent to a pH of 7. What if we have a concentration of H plus ions that's 10 times higher? Well, 10 times more than 10 to the negative 7 is 10 to the negative 6, or written out, that's 0.000001. So that's a whole lot more hydrogen ions, right? That's 10 times more. That pH is going to be pH of 6. And you notice that's the exponent, or it's the negative value of the exponent of that concentration. So 10 to the negative 6 molar hydrogen ions is equivalent to a pH of 6. Now let's look at our basic solution. Basic solutions have fewer hydrogen ions. So if we have 10 times fewer compared to the neutral solution, instead of 10 to the negative 7, that would be 10 to the negative 8. So that's 0.00000001 moles of H plus ions for every liter. That's equivalent to 10 to the negative 8 which means it has a pH of 8. So that's why those pH values sort of seem backwards, right? It's why a high concentration of hydrogen ions results in a low pH. It's because of that log function. So if you can think about it as the exponent, right, as the negative value of the exponent, that might help you understand why the pH scale has smaller numbers are acidic and larger numbers are basic. All right, that's where we're going to leave off for today.